Today, my guest is Victor Navarro. He's associate professor at Harvard Medical School, where he's on the faculty of Harvard MIT Health Sciences and Technology program and the Harvard program in neuroscience. He's an expert on the regulation of the control of reproduction by the brain, and also the interactions between energy metabolism, that is, for example, calorie intake, obesity, kind of your metabolic status, and reproduction. And a lot of this has to do with nerve cells in part of the brain, kind of a primitive part of the brain called the hypothalamus. So Victor, and also this has a lot of implications for disorders of reproduction and even enhancing sexual drive. So Victor, why don't you're from Spain originally, is that right?
Hello, Mark. Yes, I'm from Spain. But first of all, thank you for inviting me to be part of your podcast. It's truly an honor. So yeah, I'm from Spain. I grew up in the South Spain in Cordoba, and that's where I went to college and grad school, actually.
And were your parents interested in science or medicine?
No, not at all. I was the first one in the family to even go to college. So I always had an interest in science, in animals growing up. I had all kinds of pets at home. Even before I knew what genetics was, I was crossing birds of different colors as parakeets to try to get different colors. So you can call me like a nerdy kid at the time. So I always had that drive. I wanted to be a zoologist growing up until I went to college, and then I realized that making a life out of being a zoologist was not easy. Being a field biologist has a lot of problems in terms of finding a place to work, finding and whatnot. And at the time, I got interested in cell biology. I run into really motivating professors at school that brought me into that field of molecular biology and cell biology, and that's when I started working on some other studying in deep pituitary. And from there, I moved to the brain. And so when I finished college, I moved to a different school within the same city, and I started working with endocrine disruptors. So you see all these chemical compounds that are in the environment and how those endocrine disruptors were affecting the brain, and mostly the hypothalamus that you were mentioning earlier, and how the hypothalamus can be wired in a permanent way, depending on how and when the brain is exposed to these chemical compounds. So that was my beginning of grad school until kisspeptin came along, and then everything shifted to that area.
And so what is kisspeptin, and when was it discovered? How was it discovered?
So kisspeptin is produced by different groups of neurons in the brain. But the interesting thing about kisspeptin is that it was discovered in the 90s by cancer biologists. So it was an anti-metastatic peptide. And the funny thing, do you know what's called kisspeptin? Why? It was discovered in Hershey, Pennsylvania, and those investigators thought it was funny to call the name of the gene KISS1, and the peptide kisspeptin. And funny enough, they didn't know that peptide was going to be so important for reproduction. So it was very fitting that it had that KISS name and then be related to.
So it's named after the Hershey's Kisses, the candy.
That is right. That is right. And like I said, funny enough, it was very important for reproduction. So as you can imagine, there were lots of funny jokes about that in the literature. If you Google names of articles and reviews, there's always some kind of funny correlation of the name with the actual function of the gene and the peptide.
But then what led to its discovery of its role in reproduction?
So in 2003, there were two different groups internationally, one here in Boston and the other one in France. And they had access to these consanguineous families. And they had these patients that reported to the clinic that never went through puberty, that they were infertile, and through some exon sequencing, they realized that they had a loss of function mutation in a gene called, at the time, GPR54. And GPR54 turned out to be kisspeptin receptor. So this is back in 2003. They even had like really nice publications describing this and some first animal models knockout for GPR54 in mice that replicated the human phenotype. So at that time, 2003, 2004, I was in the early stages of grad school with my mentor, Manuel Tena, who is now one of the most important, if not the most important, neuroendocrinologist in the field of reproduction. And we said, well, this is so interesting. This kisspeptin must do something in terms of regulating the whole reproductive axis. Why don't we study it? And our animal model at the time was the rat. So we started with very easy experiments, just injecting kisspeptin and seeing what was happening in those rats. And we saw that there was the most potent thing that we have ever seen in terms of stimulating the whole reproductive axis. And you just,
Victor, so you said injected, do you give it intravenously or into the brain?
We gave it inside the brain and also peripherally. When we do it peripherally, we usually do it intraperitoneously, the IP.
Right.
And in both cases, it was very, very strong, very robust in terms of the stimulation that it induced of the HPG, the hypothalamic pituitary gonadal axis. So nothing was known at the time, and we decided to shift the focus of my graduate work into understanding what's going on with this kisspeptin, how was it working, like where was it working.
Well, let me go back to these people that had the mutations in the gene encoding kisspeptin that led to they didn't have puberty and infertility. So we have two copies of every gene. We get one from our mom, one from our dad.
That's right.
So these people must have had, both their mom and dad had a mutation, because otherwise they'd still have one good kisspeptin gene.
That's correct. Yeah, they were consanguineous families. So these like cousins marrying cousins, and that's how these kind of recessive mutations come up. So these mutations receptor then obviously surface when that consanguineous interbreeding happens, and that's how they were identified.
Yeah, because that would be strongly selected against an evolution, because they can't reproduce if they have.
That's right. Yeah.
So that's interesting. Okay, so kisspeptin then it's acting in the where's it acting in the brain?
So kisspeptin, let's start with where is it produced. So there are different neurons where it's produced, and the more we know, the more populations come up. But let's call it classic populations are two, and one is located in an area of the hypothalamus called the arcuate nucleus, and that is pretty much similar, the same in males and in females. And then there is another one in the AVPV, or the anteroventral periventricular area. And that one is mostly exclusive of females. And the reason for that is because that one drives the ovulation, the pre-ovulatory LH surge. So without those neurons, there's no ovulation. They need to be triggered. That's part of what is called the positive feedback of sex steroids. The other one in the arcuate, what it regulates is the pulsatile release of GnRH. And that is completely essential. Without pulses of GnRH, there is no activation of the reproductive axis. And actually, that is the hallmark of puberty onset. Puberty onset happens because we go from a period of quiescent GnRH release, there's pretty much no activity, to very strong pulses of GnRH, which, by the way, that's another topic that we can touch upon. To date, we don't know what triggers puberty onset. That's like a big black box that we're still trying to untangle. So those two populations release kisspeptin, one in the sort of surge mode, the other one in the pulsatile mode. And in both cases, those neurons target GnRH neurons. So GnRH neurons, ever since they were, or GnRH was identified in the 70s by Guillemin and Shally, we knew very little. We knew that GnRH was released in a pulsatile manner. Those were Nobel studies in the early 80s. But GnRH neurons were very few, very scattered in the brain, and they didn't seem to have any receptors that were sort of allowing these neurons to respond to factors that we now control reproduction, like metabolic factors or even sex steroids. So what happened is that when we identified these kisspeptin neurons, we all of a sudden have a very strong candidate to serve as the master regulator of this reproductive function, because these KIS1 neurons had receptors for pretty much anything that we were looking for and that we had been looking for 30 years. And
so Victor, let me kind of summarize. So you have these kisspeptin neurons, and they communicate with neurons that produce the gonadotropin releasing hormone, GnRH.
That's right.
And then that's released onto cells in the pituitary that produce the luteinizing hormone, the LH. And then the LH goes in the blood down to the gonads. And in the females, in both sexes, it promotes, I guess,
production of sex steroids and
production of
yeah,
like egg and sperm ultimately.
That's right.
Now these you mentioned these, you get a surge of LH and a pulse of NH of LH, and that the GnRH neurons alone don't seem to be responsible for that. And then you described ovulation, but what about males? Is this also happening in males?
Yeah, so the tonic release of GnRH, by tonic we call it like pulsatile part of GnRH release, that is pretty much the same. That's what it's called, the negative feedback of sex steroids. And that's the same in males and in females. You have to have pulsatile release of GnRH, and those pulses reach the pituitary, and that induces pulses of LH. And those pulses of LH induce maturation of the gonads, production of sex steroids, like you said, gametogenesis, and that is the same for both. So if you think of development, like I said earlier, during childhood, these neurons are not very active or at all, and then they get sort of awakened at some point to induce the maturation of the whole HPG reproductive axis. The other part, the ovulation part, is the one that is obviously specific of the females. And males have very few of those neurons. We don't even know if those few neurons have any biological function, or they're just there because they're kind of vestigial from evolution. Yeah. But this whole axis, the interesting thing about the axis is not only it regulates the production of sex steroids later on during postnatal life, but those neurons and those neuronal circuitries are very sensitive to the organizational effect of sex steroids. So it's kind of like a bidirectional sort of regulation. Yeah. Early on, they're very malleable, they're very sensitive to the environmental, the sexual milieu, and that determines how the brain is sort of wired permanently, which is a very interesting topic that we also study in the lab. How the brain develops into masculine or feminine, and how that depends on how much of a specific hormone you are exposed to during development in the womb.
Okay, so the estrogen and produced by the female gonad cells, and the testosterone by the male, they travel through the blood up to the hypothalamus, and they don't, from what I gather, they don't act directly on the GnRH producing neurons, but they are acting on the kisspeptin neurons. Is that right?
That's correct, yeah. For the most part, because GnRH neurons have, there's always a little bit of controversy about this, but we know that GnRH neurons have estrogen receptor beta, and there's been some electrophysiology studies showing that estradiol may be involved in the way that GnRH neurons respond. But overall, different studies that have been performed on removing estrogen receptor alpha, which is the one that plays the most part of this regulatory role, happens at the level of KIS1 neurons, both in the arcuate and in the ABPV. And like I said, there are other neurons in other parts of the brain. For example, we are now studying KIS1 neurons in the amygdala. We think they're involved in sexual behavior. So other populations are coming up, but sort of the classic are these two, and they respond directly to sex drugs, like you said. Not only estradiol, but also testosterone, because they have androgen receptor as well.
And okay, so let's talk about the links between energy metabolism and this neuroendocrine system that you've been talking about that's controlling reproduction. So animals in the wild, if they're kind of in essentially a starvation condition where there's not much food around, the females will stop cycling. Their estrous cycles will stop, which kind of makes sense, because you don't want to, become pregnant and have to put a lot of energy into the growth of the babies when there's no food around. Actually, I had a postdoc, Bronwyn Martin. She's now in Belgium. She's from Belgium. And she did some experiments in rats. She had males and females, and she put them on, she had 4 different conditions with regard to energy intake. Ad libitum feeding, so that is the animals had food available 24/7 every day. And then she had a group with 20% reduction in calories below their normal intake, a group with 40% reduction, and then a group that were on every other day food deprivation or every other day fasting. And she let them go for, I think it was like 6 months. And in the females, she did like vaginal swabs, I think, and kind of determined whether, bottom line is she determined whether there's cycling. And then at the end of it, she killed the animals. She looked at their gonads. She did a lot of stuff, looked at their brains. She also did some behavioral testing. But there's a couple interesting differences between males and females. The females, when they're on like the severe calorie restriction, 40%, they stop cycling. And interestingly, they became more physically active, and they lost pretty much all of their fat.
Right.
Or at least their abdominal fat. The males didn't lose as much weight. They didn't become like hyperactive, and their sperm count was normal. So kind of our interpretation of that from an evolutionary perspective is, again, if you're under kind of starvation conditions, if you're a female, you want to stay alive. You don't want to get pregnant.
Yeah.
But if you're a male, maybe you want to impregnate as many females as you can before you die from.
Yeah, exactly. You got to think about it in the context of who carries the main burden of reproduction, at least in mammals, and that's the female. And we see that in the lab, that the sort of circuitry mechanisms that regulate reproduction in the female are much more controlled, like much more tightly in the female. Very subtle modifications of the metabolic environment affect reproduction in the female. And males, you pretty much can do whatever you want. That it's really hard. You have to really hit them hard with a starvation protocol for them to become infertile. And it also makes sense what you were saying about increasing activity, because you're activating food-seeking behavior neurons like AGRP neurons. And that's part of why it was, I guess, evolved that way to increase the interaction between neurons that control feeding behavior and caloric expenditure and reproduction. They're both located in the arcuate. I mean, of course, the brain is big, and there are other neurons that contribute to the regulation of these neurons in other parts. But if we think of the main regulators of food intake, those are these AGRP neurons and these pro-opiomelanocortin neurons. They're intermingled with these kisspeptin neurons, and they talk to each other. So this is how the regulation happens. And if we step back to the introduction that you were given, it does make a lot of sense, because in the wild, like you were mentioning, you don't want a female to engage in reproduction if she doesn't have enough reserves to even maintain herself. So in a way, if you think about it, the neuroendocrine control of reproduction is a great proxy for overall health of any animal. If you don't have enough energy reserves, it shuts down. If you're stressed, it shuts down. So a good, marker of overall health is how is the reproductive function working in this specific animal. So this is the reason why it evolved that way, which interestingly, for the species is the opposite. In terms of perpetuation of the species, reproductive function is absolutely essential. And this is the paradox that we are seeing, that if you want to modify any of the sort of central players that control reproductive function, for example, knockout mice models, you knock out a gene, and then you try to look at what happens when you don't have that gene. The possibilities for compensation are huge, because throughout evolution, there's been a lot of circuitry that has evolved to make sure that if something goes wrong, you might not be the most fertile, but you still can reproduce. So that's what, that there's an evolutionary advantage of that. However, at the individual level is the opposite. You just want to make sure you survive.
Yeah, and in the group we had that did the intermittent fasting every other day, food deprivation, the females, they kept cycling, but there was some, not major, but detectable irregularities in the periodicity of the cycles.
Right.
And in the wild, a lot of animals, they eat in an intermittent manner.
That's right.
So that's kind of interesting.
Yeah.
So have there been studies done in animals to look at the effects of energy restriction on kisspeptin neurons?
Yeah, absolutely. And those are actually some of the studies that I started working on in my graduate work with rats at the time. And we were seeing that indeed kisspeptin is very sensitive to the environmental, I wouldn't say environmental, but to the energy reserves of the organism, the rat in that case. So when we were doing caloric restriction, like the one that you're mentioning, or pure fasting, or even diabetic rat models, which pretty much in a way are, calorically restricted, or leptin deficient mice, there are different models that you can consider, a good model for caloric restriction. We saw that kisspeptin neurons were inhibited. The expression of the gene was really decreased. An interesting thing is that by giving exogenous kisspeptin, we were able to rescue this. So one of the ways that we saw it very clearly was by submitting these animals to caloric restriction before puberty. So we hypothesized that if Kiss1 neurons were so important for the activation of the axis, they should be involved in puberty onset too. And if the animal doesn't have enough fuel, those neurons wouldn't be active, and therefore the animal wouldn't go through puberty. And that is exactly what we saw. And we can rescue it by exposing the mouse to a exogenous treatment with kisspeptin. So these are approaches that right now are being tested in the clinic, because there's different mutations that lead to hypogonadotropic hypogonadism in kids that never go through puberty. So kisspeptin, using that approach from the rat, is now being used first to test if the individual has a developmental problem at the level of GnRH, for example. And if that were the case, the child wouldn't respond to kisspeptin because the GnRH down would be dysfunctional. But if you see a response to kisspeptin, you can say that this kid is eventually going to go through puberty. He or she might be just a little bit delayed. So it's being used as a treatment, but also as a diagnostic tool.
And so there's a lot that happens with the calorie restriction. Glucose levels in blood tend to stay kind of low normal range. Ketones go up. Leptin is down, ghrelin is up. And by the way, I had Alan Watts in a previous podcast who talked about the control of eating behavior. So he talked about leptin and ghrelin and so on. And so is there any evidence that either the kisspeptin neurons or the GnRH neurons respond directly to
leptin, for example?
Yeah, or even energy substrates like ketones or glucose?
Yeah, so in terms of leptin, this is something very interesting because Kiss1 neurons do have leptin receptor. GnRH neurons don't. And that sort of further elaborates on what I was telling you earlier, that those neurons were just like there sitting, waiting for something else to tell them what to do. But Kiss1 neurons do have leptin receptor. But the interesting thing is that there's been studies removing leptin receptor from Kiss1 neurons, and there wasn't really a phenotype in those mice. So what we and others did was to look at upstream neurons, because what we know is that the action of leptin to regulate both metabolism and reproduction happens at the level of the brain. Even though there's leptin receptor in other parts of the brain, you get an animal that doesn't have leptin receptor, you put it back in the brain, and the animal is pretty much normal. So that tells us that it happens in the brain. So the next question that we had was, well, where in the brain is this happening? So a first broad approach was to look at glutamate versus GABA neurons. And so we did this, we removed leptin receptor in either population, and we saw that the action was happening through GABAergic neurons. And that was the same as the mechanisms that control food intake. So there are some upstream neurons to kisspeptin that are GABAergic and that we still don't know which ones they are, but we know that they're upstream and GABAergic, and they transmit the information that leptin sends to the brain.
So with obesity, often there's what's called leptin resistance. These neurons in the brain it's kind of like insulin resistance.
Yeah.
Right.
Yeah.
The neurons don't respond to the hormone. What happens with obesity is it's a huge it's an epidemic essentially in this country, including childhood obesity.
Right.
And this what you're describing on these neuroendocrine systems and neural control suggests that there may be some roles for abnormalities in the system in children,
right,
and adults with obesity. Could you talk about that?
Yeah, no, absolutely. In fact, if you look at the statistics, the age of puberty onset is advancing quite significantly. If you look at data from early in the 20th century to now, it's advanced quite significantly. And one of the reasons for that is that obesity in children has increased. I mean, not only obesity, just like nutrition in general has improved from a century ago, but in the last few decades, also childhood obesity is becoming a big problem. And that additional sort of activation that fuel the excess energy reaches the brain and sort of promotes and induces the whole machinery that induces puberty, that is there, and we know it's happening, it's becoming activated. And that's a big problem because I mean, you don't want to have advanced puberty because that can induce a lot of physiological and psychological issues. So one of the reasons why we're so concerned about childhood obesity, not only other health outcomes later on in life, is the significant advancement in the age of puberty onset. And if you move that to adulthood, then you get into what you were mentioning, the leptin resistance, for example. And that's an interesting topic too, because if you remember when leptin was identified kind of like 25 years ago or so, it was considered like it's gonna solve the obesity problem. It's just gonna be great. We're gonna inject leptin, and all of a sudden people are gonna stop eating, and it's gonna be, you know, great. And it hasn't happened. And it makes sense that it hasn't happened if you go back to how evolution works. Throughout evolution, what was a problem was if you didn't have enough energy. Throughout millions of years of evolution, there was never the too much energy considered a problem. Yeah. So if you don't have energy, obviously leptin, the absence of leptin signaling is a trigger for you to seek food, shut down reproduction, and be very effective at survival.
Yeah.
If you go to the other end and you have a lot of adipose tissue releasing lots of leptin, you get resistance because your body is not going to act against that. I mean, evolution has designed us to be okay with that because in theory, other times with no energy will come and you will need that. So that's the underlying reason why those initial theories about leptin as a treatment haven't worked.
And what do you think there's some evidence, right, that with obesity, there's actually inflammation in the hypothalamus, and that the inflammation could be contributing to this leptin resistance?
It is very likely. You know, with obesity, you have all these interleukins and many other factors that are released, and we don't particularly study that. And to my knowledge, there hasn't been any clear study addressing how kisspeptin neurons are sensitive to inflammation. There's been some protocols using LPS, you know, it's kind of like a pro-inflammatory molecule, sort of showing that indeed inflammation, anything that is sort of bad for you, is going to shut down KIS1 neurons. And we go back to what I was mentioning earlier, that the reproductive axis, mostly at the central level, is a great proxy for overall health. So if you have chronic inflammation and that's reaching the brain, more likely than not, you're going to have an inhibition of the KIS1 system. So, but the whole mechanism how this happens, we don't know yet.
And so you mentioned the GABA, the inhibitory neurotransmitter GABA. And I had a postdoc who did some studies where he put mice on every other day fasting or control, and then he made brain slices and recorded from individual neurons in the hippocampus. We had a lot of interest in the hippocampus for many reasons. And so the bottom line is he found that not immediately, but over a period of several weeks, following the onset of intermittent fasting, there's an enhancement of GABAergic tone, which kind of quiets the circuitry in the hippocampus. And do you think there's any role for enhancement of GABA in the effects of energy restriction on this system?
Yeah, absolutely. And that is a really good approach and a common pathway that gets activated when we don't have enough energy. And the first neurons that come to mind are AGRP neurons. Those AGRP neurons are GABAergic, and they're called hunger neurons because they're activated when we don't have enough energy, and they promote feeding behavior, and they do it in both ways. One is through the binding of AGRP to the melanocortin receptor 4, and that is considered a sort of slow-acting mechanism. So over time, through that mechanism, you increase food-seeking behavior. But then there's another mechanism that is quick, that is going through GABA. So if you start a fasting protocol, for example, that's an acute sort of model, and you know that when you're hungry, when you haven't eaten for, let's say, 8 hours, it's quick. You have that drive to find food, you're very hungry. And that fast-acting mechanism goes through GABA. So those AGRP neurons are sitting there in the basal part of the hypothalamus. They project to many different areas of the brain. I'm not sure about the hippocampus, but I would bet that they do too, because they're far-reaching. So whether that is the case or not, I can't really speak to that. But what I'm telling you is that GABAergic circuitries in the brain are known to be active when there is a negative energy situation.
Well, that's interesting. So, typically we think of the GABA interneurons as, their axons stay within a very localized region. Maybe they impinge upon 100 neurons, excitatory neurons right around them. But you're saying these GABAergic neurons in the AGRP neurons have axons that go outside of the hypothalamus.
That's right. Yeah, they go at least to make different nuclei within the hypothalamus, and because that's the main area that we are interested in. I can really give you a name of other areas outside of the hypothalamus, but I would bet you that they do.
I bet you they go to the hippocampus. It makes a lot of sense because the hippocampus is involved in spatial navigation.
That's right. So
right, and if you're
food-seeking behavior,
if food-seeking behavior, and remembering where you've been and what was there previously, and so on.
Absolutely, yeah, yeah. And the interesting thing, this is not our work, but these AGRP neurons show anticipatory behavior. Just like showing food to an animal is enough to change the activity of the neurons, because sort of predicts that you're going to have food intake. So they start regulating the whole, food intake and energy expenditure behavior before that, so you don't overshoot later on in terms of eating too much or too little.
Yeah, so far you've talked about studies in animals, and the reason's clear. It's you can invasively interrogate their brains and hypothalamus, and you can do genetic manipulations of the receptors, or you can infuse kisspeptin and so on. What about humans? And in a lot of brain regions, people use imaging, functional imaging, MRI, to look at, are neurons active and to what extent under such and such conditions. And you mentioned, showing food to an individual and seeing effects. Is anybody doing studies in humans trying to,
So the main the main area where studies in humans are being performed in terms of brain imaging that you're mentioning relates to sexual behavior. So I mentioned earlier that there is another population of kisspeptin neurons that is located in the limbic system, in the amygdala. And interestingly, this is a work by Logitello and Alex Cominas out in London. And it's very interesting because they treat patients that have some sexual dysfunction in terms of very low libido they expose them to kisspeptin, and now they show them different images, some neutral, some aversive, some of sexual content, and they see that kisspeptin really highly activates that part of the brain, the amygdala, and induces some sort of like sexual drive. So that's been proven in men, and I know there are studies now working on testing whether this is true in female. So it's a very promising area. It's not still in the market, but there was a lot of buzz in the news a couple of years ago. They called it the mental Viagra, in terms of being able to mentally stimulate people to engage in sexual activities. So it seems like kisspeptin not only induces the activation of the reproductive axis in terms of inducing sex steroid release, but also behavior, and it makes sense. There's the same molecule doing both things. Yeah.
Now you made me think of addiction. Now, so people can become addicted to food, particularly highly palatable foods, sweet foods or fatty foods, and
that's right.
It's been shown that there's what happens with the dopaminergic system in people with chronic obesity is very similar to what happens to a drug addict. So what you said about kisspeptin and sexual drive made me think it may cause someone who is or otherwise kind of prone to becoming addicted to pornography
probably
to become, it could be end up that they're
there's connection from the system to dopamine neurons, mostly in the whole preoptic area, and those dopamine neurons control sexual behavior. That's been shown recently. So
there's potential benefit clearly for people who are having low libido and low sex drive.
Right.
I could see some potential side effects.
Yeah, of course. So I don't know where that's going to end up, but I know there are some early phase clinical trials right now going on, and it'll be very interesting to see what happens, because it's a big market for that. So pharmaceutical companies have a lot of interest on that aspect of kisspeptin, out of the many aspects of kisspeptin, because it seems to be involved in many different physiological functions, not just reproduction itself.
Yeah. So you've talked about hypogonadism, and you talked about sex drive, but there are several other disorders. So anorexia nervosa, which usually affects young women, and they don't eat, they tend to exercise a lot, and their estrous, they quit having menstrual cycles. And then there's amenorrhea and polycystic ovary syndrome. Do you want to say anything about those three?
So those are sort of one of the main syndromes that we know that are happening mostly on females, and the anorexia nervosa is a very good example of what we were saying before in the wild. If you don't have enough energy, so this is taken to today's world, what happens unfortunately in many, mostly teenagers and mostly girls, that they restricted their eating, they overexercise, and the first thing that happens to these girls is that they lose their menstrual cycles. And this is the same kind of response. These kisspeptin neurons are responding to the lack of enough energy by shutting down the whole axis, just preventing that by any chance you would get pregnant at all. So that's that part. And then polycystic ovary syndrome, PCOS, is another is a very disturbing disease for women that suffer this, and it's actually a lot of women. It's like up to like 10 to 15% of women. An interesting thing about PCOS is that one of the hallmarks is increased pulsatility of LH. And if you remember what we were discussing very early on in our discussion, KIS1 neurons in the arcuate are driving these pulses of LH. So there's a few labs around the world, and ours is one of them, trying to understand whether these KIS1 neurons are actually dysregulated because of the exposure to high levels of androgens and probably other hormones like AMH throughout development. And whether that disruption of these KIS1 neurons is leading to these increased pulsatility of LH, which is what drives the hyperactivation of the ovaries. And because of that hyperactivation, you have an increase in androgen release, and it sort of feeds back. Those androgens go back and maintain that feedback loop. So what we're trying to do now is to tackle those KIS1 neurons.
You said androgen, so which testosterone, right? Which cells are producing it in the female?
So in females, the overactivation of the theca cells in the ovary can release androgens because of the constant hyperstimulation by gonadotropins. That's to my understanding. I mean, I'm not an OBGYN, but
then do those women have any signs of masculinization?
Yes, they have hirsutism, which is the increase in facial hair, for example. They have oligomenorrhea, which means they have few menstrual cycles. I think it's considered less than 8 menstrual cycles throughout the year. It's considered, I believe, oligomenorrhea. And they have problems to conceive. They have lots of fertility problems. So in a way, they do show signs of masculinization because of this exposure to high levels of estrogens. So androgens, sorry. So what we're trying to do now is to inhibit those KIS1 neurons and see whether we improve the conditions of those women. So we have some promising data in the lab. We have mouse models of PCOS, and they reproduce most of the hallmarks of polycystic ovary syndrome women, which is high androgens, low estrous cycles in this case, and disrupted LH pulses. So when we inhibit these KIS1 neurons, we can rescue most of these parameters. And so there's some promising evidence that KIS1 neurons could be a therapeutic target for PCOS in the not too distant future.
And in the animal model, is it a gene mutation, or is it some other way to?
The animal model that's a great question. The animal model that we use is a prenatal androgenized model. So what we do is we get the dams, get the mothers when they're pregnant at the late states of their pregnancy, and we treat them with DHT, dihydrotestosterone, between day 16 and 18. And then the female offspring is masculinized in that way. It's showing signs of PCOS later on in life. So we don't have to add any other treatment, because the different treatments for PCOS is another one that uses letrozole. There's several ones, but those require manipulations later on in life. This one, the animal postnatally has no manipulation, and the females show those signs of androgenization, so to speak, which we think is more faithful to what happens in women, because women that are daughters of mothers with PCOS have a higher chance of developing PCOS because of the exposure in utero to high levels of androgens, high levels of AMH.
So that now you're making me think of epigenetics, right? Where there's changes in the expression of gene, that is essentially the amount of protein
Correct.
produced from that gene, but there's no genetic defect. And I talked about this in a couple of previous podcasts, the notion that what a parent, particularly the female, what a mother, her environmental conditions or metabolic state during pregnancy can affect a lot of things in the offspring, whether or not they develop obesity, their behavior. Could you talk a little about, I know you're interested in epigenetics.
Yes.
Can you talk about this?
That's a fascinating topic too. Epigenetics is something that is less known by the common public. Basically, what it is the interaction between the genes and the environment. So there are certain conditions that determine changes in the genome through methylation or through histone modifications or non-coding RNAs that lead to permanent changes in the genome. An interesting thing, something that most people don't even think about, is that this can be transgenerational, meaning that you're exposed to some kind of insult. And when I say that, I'm talking about a long period of famine, for example, low energy intake, that induces changes in your genome that you can pass down to your offspring and their offspring to the next one. So it's really rare to track this. There's been some animal models that you can show that indeed this happens. The most common scenario is what you were talking about, that exposure during pregnancy to the offspring. And if you think about it, you're targeting 2 generations there, because you're targeting the fetus and you're targeting the germ cells of the fetus that are already there. So that's direct exposure to 2 generations. And whatever conditions happen to the mother in that state, whether they're exposing the fetus through the placenta to high levels of androgens or metabolic aberrations, too much or too little, that can induce changes in this, what I call this methylation or histone modifications in specific genes. And you see changes that are maintained throughout the life of the offspring. So children of obese mothers, or even obese children, because that developmental phase is still sensitive to epigenetic changes, induce some sort of pattern of expression of those genes are going to be maintained. So that might lead to permanent obesity later on in life, reproductive disorders later on in life, a whole array of endocrine problems that derive from that early exposure, either to you as a child, during pregnancy in your mother, or even other generations. So that's a fascinating area.
Yeah, there's thinking about the accelerated onset of puberty caused by childhood obesity and the potential for that being transgenerational. That is to say, for example, say a woman has childhood and adult obesity and she has children, that child may be more prone to obesity, but say they're not. Say they eat a healthy diet, don't overeat, exercise. Nevertheless, their children, like the second generation, may be even prone. Now, one thing I was thinking about is, with accelerated development, is in autism, there's a lot of evidence that there's accelerated growth of the brain during embryonic and maybe even early postnatal development. And there's, what's been clearly shown is there's hyperexcitability of neural networks. For example, autistic kids have increased incidence of seizures. Do you know if anyone has looked at onset of puberty in children with autism versus not?
That's an interesting topic To my knowledge, no one has looked at that, or at least I haven't really encountered that data.
Okay.
That's an interesting topic because puberty onset is sensitive to multiple other diseases. For example, some time ago, we were interested in traumatic brain injury. So children that have suffered traumatic brain injury have a higher ratio of advanced puberty. And I was interested in knowing why that was. And there's a number of molecules. One of them is substance P that is induced by trauma. And we've seen in our lab that substance P is part of a group of proteins called tachykinins. And tachykinins are known to be an important piece of all these machinery that I'm telling you about kiss-1 neurons. They're actually essential for kisspeptin pulses to happen. So we haven't directly tested that, but there is, like I said, evidence that traumatic brain injury induces and increases substance P, and then we have shown in the lab that substance P induces puberty onset. So there's likely a correlation there. So whether the same situation happens in autistic kids, if they have increased levels of, for example, substance P, could very well induce precocious puberty.
Yeah, I forgot to mention one important thing there, that there's epidemiological studies have clearly shown, and animal studies bear this out, that there's increased incidence incidence of autism in children born to mothers who, if they have obesity and or type 2 diabetes during pregnancy. And so, you know, presumably, since they're more prone to, the child is probably going to be more prone to obesity and maybe more likely to have a accelerated onset of puberty. So that would be interesting to look at.
It's very interesting. No one has looked at this in kiss-1 neurons, but if we go back to those AGRP neurons that I was mentioning, that has been looked at not in the context of autism, but in the context of early manipulation of energy environment. Too much or too little energy reserves can affect how those neurons develop in terms of axonal length and synaptic contacts that they make. So it is very likely that other neuronal groups are affected as well.
Yeah. My mind's kind of racing here. So there's one thing I forgot to ask you. We've covered a lot of ground, and like maybe I'll have you summarize and tell our listeners and viewers what you're doing now and what you think the major unanswered questions is. But one kind of specific question before we get to that. Has anyone looked at whether ketones, beta-hydroxybutyrate, acetoacetate, affect this system, the kisspeptin, GnRH, LH system?
The short answer is no. No one has really looked at that, at least not in a detailed manner.
Because they do have signaling. They do have signaling functions.
That's right. Yeah.
You know, there's Eric Verdin has shown they actually affect gene expression by modifying histones. And we found that beta-hydroxybutyrate, which is elevated during fasting, will increase BDNF levels, brain-derived neurotrophic factor. BDNF actually has a anorexic effect. It will inhibit food intake. Do you know if anyone's looked at whether BDNF affects this system?
No, not that I can recall right now. Yeah, there are not that many groups looking at metabolic regulation of kisspeptin neurons. So I feel like I would know. I hesitate to say 100% no, but at least know that I'm aware of.
If you knock out both alleles of the BDNF gene, the animals die during fetal embryonic development.
Yeah, that I know.
But if you knock out one allele, they develop obesity and diabetes.
That would be very interesting to look at.
Yeah, that would be interesting. Very interesting. Yeah.
Yeah.
And it's produced in an activity-dependent manner by a lot of neurons. I would be very surprised if there aren't neurons impinging upon or maybe even kisspeptin neurons themselves produce
Right. Well,
estrogen.
maybe
So,
there's something that we need to talk about more in the future, Mark.
yeah. Okay, so what's your most exciting questions you're trying to answer now in the lab? And why don't you describe your lab up there in Boston?
So my lab is a relatively small lab. We're around 10 people. So I mean, I guess it's a decent size. And we have different projects that all sort of bring together different parts of neuroendocrinology, but all related in a way with reproduction. So we continue to try to understand what drives puberty onset. That's been a driving force for the lab for some time. But in addition, we continue the study of the melanocortin part of sort of melanocortin part of regulation of metabolism and reproduction. So that's the main line that continues this metabolic regulation of reproduction. We're finding very interesting things there in terms of the action of melanocortin receptor 4 within KIS1 neurons that regulate ovulation in females, and that also in males trigger changes in body weight. We also have a line that focuses on thermoregulation. This is something that we haven't touched upon, but it's very, very interesting, which is that these KIS1 neurons are considered now the triggers of hot flashes, menopausal hot flashes.
Oh.
And makes a lot of sense, because when a woman goes through menopause, what happens is that ovarians get sort of depleted in their ability to release estrogens, right? And then these KIS1 neurons in the arcuate that I was telling you about earlier, they don't know that menopause is a thing. Throughout evolution, if we go back to that, very few, if any, women ever reached menopausal age. So now we get into a situation that those neurons are not sensing circulating levels of estrogens, and they're trying to compensate, which is not going to work because the ovaries are, just like reaching their sort of reproductive life. And the hyperactivation of those neurons is inducing hot flashes, because in addition to targeting the GnRH neurons that we talked at the beginning, they also target thermosensitive neurons in the preoptic area. So there's a lot of research from my lab and others, and interest from pharmaceutical companies trying to target these KIS1 neurons, inhibit them, and when you do that, you significantly improve the number and the severity of the hot flashes, which in essence what a hot flash is the right term is vasomotor symptom. You get vasodilation of the capillaries around your periphery, and increase in heat dissipation. So that's why women feel hot. And I have to say that men can also experience this. It's just under different conditions, but men that go through androgen deprivation therapy for prostate cancer, for example, also experience this. Or even kids that are treated with a GnRH analog for gender dysphoria can experience this. So, but in any case, what happens is that you get this vasodilation, you have that heat sensation because of the heat dissipation that leads to the hot flash. So all that is triggered by these same KIS1 neurons in the arcuate. There's a lot of interest in that area now. And we have another line in the lab that is focusing on sexual behavior, targeting those KIS1 neurons that I mentioned in the medial amygdala. We're trying to understand whether these neurons are connecting behavior and activation of the axis. And this is critical for reflex ovulators, for example. Those ovulators that ovulate when there's copulation. And mice are not considered reflex or induced ovulators, but the mechanism is there, and we believe that under certain conditions, copulation can trigger ovulation in the mouse too. There are many species, like rabbits, cats, that only ovulate when there is engagement in sexual behavior. However, deconnection, how behavior is connecting the actual release of gonadotropins to ovulate is not known. So we think that these kiss neurons in the medial amygdala might be involved in playing that role. And I'm not saying that they are, I'm saying that we're testing that hypothesis now. So that's another main project. The final one is related to stress. I mentioned earlier that stress, and we all have experienced stress at some point in our life, can affect reproduction. And that's a leading cause of infertility is the high incidence of stress. Now, so we're trying to understand how stress is affecting the whole activity of the reproductive axis. And when we look at older females, how stress can also affect the severity of these hot flashes, because everything goes through the same sort of group of neurons. So those are very generally the areas that we're working on in the lab.
And so I did a podcast with Robert Sapolsky on stress. And so, of course, there's a neuroendocrine system that controls stress. You've got CRH, corticotropin releasing hormone, and then it causes release of adrenal corticotropic hormone and causes the adrenal glands to secrete cortisol. So do the kisspeptin neurons, I would assume they have cortisol receptors.
They do. They have glucocorticoid receptors, and that's one of the experiments that we are doing right now. Actually, we're trying to remove glucocorticoid receptor from kiss-1 neurons and look at the reproductive phenotype and the thermoregulation ability of these mice.
A lot of studies of stress in animals, they will remove the adrenal glands, remove the source of the glucocorticoid. In animals, if that's done in females, does it affect do you know if it affects their reproductive cycle?
I'm not sure if anyone has done that. I mean, I would guess yes. I don't, recall of any actual experiment.
I can't think of, in my mind, I can't think of seeing I used to work on that system, and I don't remember seeing anyone actually reported anything about that.
I would guess the animal is very sick anyway, so you got to be careful when the animal is feeling sick. There's a lot of different mechanisms that get triggered and that ultimately affect the reproductive axis, as we've been talking about. It's so sensitive to anything that goes wrong that
you need to knock out the androgen receptor in the KISS1 neuron, the kisspeptin.
Exactly, that's what we're doing. We're knocking out the glucocorticoid receptor there, because you cannot get a glucocorticoid receptor whole body knockout, because they're lethal. You really need corticosterone signaling.
Interesting. Yeah. Great. Well, you're in a good place now. I'm sure you got a lot of good people up there to collaborate with. So that's nice. And you'll probably even get into collaborating on human studies, I imagine, as this moves forward.
Yeah, anytime we can. It's always great to have that translational factor in any study. So it's not easy to do human research, but we're trying to get there.
Yeah, it's an exciting time in your career. You're at a stage where you're.
Oh yeah, I mean, we're very, very lucky to be working at this time. The neuroscience techniques have evolved at a speed that is really, really hard to keep up with right now. In comparing to, I went to grad school 20 years ago, and nothing that we do today was available when I was in grad school, and that's only 20 years. I cannot even imagine in another 20 years what will happen.
The cell-specific gene manipulation and the optogenetics.
Absolutely, yeah, we do all that, and the viral delivery of pretty much anything you want to. CRISPR has accelerated any manipulation enormously. All these imaging techniques, vivo, like calcium imaging that we do, it's really impressive and potent. So it's a great time to be working on anything neuroscience related.
And I can see, you've been talking about from therapy for reproductive disorders, giving kisspeptin or things like that. But you can imagine doing, targeting genetic, using CRISPR or whatever, targeting, say, only these kisspeptin neurons and not those kisspeptin neurons.
Correct. Yeah.
Something
I mean, we
like
do that. We do that already in mice.
Yeah.
But maybe someday that will, be
But
moved to the human.
as you know, there have been clinical trials of CRISPR,
Right.
mostly so far for this manipulating genes in white blood cells, but
Correct.
this is going to move towards targeting specific cells in different organs, I think.
Yeah. Eventually, we will get there. That personalized medicine that I think will be a whole new era.
Yeah. Very exciting. Well, thanks for your time, Victor. I enjoyed talking with you, and it gets me back in thinking about neuroendocrinology.
Oh, thank you so much for having me. It's been a true pleasure.
Okay.
Right.
Have good holidays.
Thank you. You too.
Okay.
Bye-bye.
Bye.